All, in this lecture, we'll see about the next type of the staining method used to differentiate your microorganisms. We call this as your special staining method. So special staining methods are used to visualize special structures like your capsules, endospore, flagella and etc. that makes bacteria easy for their classification and grouping based on the presence or absence of the listed special structures being present in bacteria. Capsular staining method. So this type of staining method is used to demonstrate the presence of capsules around some bacteria and fungi. So capsules are often made up of your polysaccharide, and in some organism it is also made up of your hyaluronic acid, specifically your Streptococcus species. So this capsules helps in imparting the pathogenicity to the particular organism and cause disease in human beings and other animals. So some of the capsule bearing bacteria are your Klebsiella species and Streptococcus species. The only encapsulated fungal species existing is called as your Cryptococcus neoformans. There are three different methods used to demonstrate capsules in the microorganism. So the first method is your negative staining method, which we use your nicrosin or your India ink. In Antony's method, we use your methylene blue, which is also called as your positive staining method. And the third reaction is your Quelling reaction, which is an immunological method used for the detection of the capsules around microorganisms. First, speaking about your negative staining method, so the procedure for your negative staining method is place a drop of India ink or a nicrosin at one end of your slide. Next, a loop full of culture is added to the drop of the stain that is kept on one end of your slide. Next, with the help of the other slide, make a thin smear by keeping the other slide at 30-degree angle on a previous slide. Now air dry it and saturate the slide with crystal violet for 1 minute. Next, wash the slide under your running tape water, blot dry or air dry it and observe under your microscope. So what really happens is the capsule appears colorless and the crystal violet that is added into the smear is taken up by the vegetative cells and your India ink or nicrosin stains your background. So a highly refractile, larger, colorless capsules are seen under your microscope, and the capsules that are seen or visualized under the microscope is the size twice the size of your vegetative cells, and it is a larger capsule being visualized under your microscope by this technique. Speaking about Antony's method of capsular staining, it is called as your positive staining method. So the procedure for your positive staining or Antony's method of capsular staining is smear the slide with the given organisms, that is basically the organism that bears your capsule. Stain the smear with crystal violet for 1 minute. So once after 1 minute, wash the slide with 20% copper sulfate solution and blot dry or air dry it and observe under your microscope. So what really happens or what we get is the crystal violet that is purple in color is taken up by your vegetative cells, and these vegetative cells appears purple in color. The copper sulfate, which is basically of blue in color, that stains your background, and we can visualize a colorless, highly refractile capsule against your blue background as shown in the image below. This is the result that we obtain by performing your positive staining method for capsule under your microscope. Is the Quelling reaction for the demonstration of your capsules. So this Quelling reaction is a gold standard technique used for the demonstration of capsules, which is specifically used in clinical diagnosis of serotypes of Streptococcus pneumoniae and other bacteria by visualizing the capsules that is formed around the particular organism. So it is an immunological method which involves the reaction of antigen and antibody. So speaking about the procedure for your Quelling reaction, place a drop of broth culture on the slide, that is the organism that has to be visualized under your microscope was made as a broth, and then a drop of culture was placed on a slide. Now add a type specific anti-sera to it. So the anti-sera is basically your antibody that is available against a particular organism, and the culture is termed as your antigen, and the anti-sera here is your antibody. Now the antigen antibody reaction is allowed to take place. So add a drop of methylene blue for 1 minute and then wash the slide, blot dry or air dry it and observe under your microscope. So what exactly happens here is, so due to the antigen antibody interactions, the capsule gets swelled up and it is twice the size of the vegetative cells, and a highly larger capsules are visualized in case of your quelling reaction. So as you can see in the picture given in the bottom, for the negative there is no capsules, there is no bulged forms around your vegetative cell, but in case of your positive side we have your bulged highly refractile capsule being present. So this antigen antibody interaction helps in increasing the size of the capsules, and so the capsules can be visualized clearly under your microscope. Due to this specific interaction or activity taking place in this type of staining method, this is considered to be the gold standard method for the detection or demonstration of your highly refractile larger capsules around your microorganism than the other two methods that we have discussed earlier.